Okay. Hi, everyone. My name is Michael Caudy. I'm a genomics researcher, and I study genetics and genomics. I've been doing that for many years. I've been using content management systems to build custom knowledge bases for genomics research and also for crisis projects. And I started a crisis project for the COVID infection, and basically, it's kind of interesting that crisis projects are similar to genomics in that they both involve extremely large amounts of data that's changing very fast. And in genomics, it's because you keep coming up with more and more sequence that you have to keep dealing with on a day-to-day basis. And in a crisis project, there is typically a very complex set of events happening on the ground at the same time. So since this is CMS Philly and you're all hardcore CMS developers or wannabe hardcore CMS developers, I'm going to try to present this in terms of the experience that I've had over the last about 20 years, taking what at different times has been the best open source content management system available, and now that's Drupal, using the Panopoly base distro, and extending that typically with some domain-specific languages. So I create a knowledge base that is extended to essentially any given knowledge domain for the problem at hand. And in this case, it's a crisis problem. So what I'm going to do is I want to start showing you slides now. And let me go back here. Where did that go? Okay. So.
Yep. Perfect. Awesome.
Okay. So. So what I'm going to do is show 3 examples of creating custom knowledge systems for genomics research and human crisis projects. And I'm going to start with the genomics and give you an idea of what can be done with this. In 2005, I developed a program called G-Clusters, and it's for whole genome transcription code knowledge analysis. And the goal is to understand, in particular, how do neural specific genes get turned on during development. How every cell has the same genes, but only certain cells turn on neural genes that make them neurons. That's what I studied. There's a DNA code in the genome for that, and that's what we're going to talk about. That's called a transcription code. So based on that kind of background, I will then present a previous crisis project that I put a lot of time and effort into in 2010 after the Haiti earthquake. And basically found that I could take the same kinds of, used a Drupal content management system with a bunch of other Drupal developers to create a very powerful data mashup, essentially, that let us get food to people who needed it and to connect them up with people who had the food. So I'll show you a bit of that. And then I'll come on to this 2020 COVID-19 crisis project and knowledge system. So the first part of my talk is 3 examples of creating custom knowledge systems for genomics research. And every one of these has involved creating what I call a custom local knowledge base, where you're storing all of the information that you want to present online, well, on your own database or knowledge system. And the purpose of that, because say with a crisis or anything else where you have a lot of rapidly changing data, there's this phrase, the river of news, that is just all of this information flowing by. And what you want to do is select out using a domain-specific language and like hashtags and stuff like that to select the data that you want, store it locally, create whatever images you need and so forth, store it locally so that you can review it very quickly. And also you can display dashboards and stuff that make it clear exactly what is important and with all of this information flowing by. So I'm going to give those 3 examples. Then in the next part, I'm going to talk about next steps for the COVID-19 crisis project and knowledge system. I need help with this Drupal project, just lots of things, design plans, and I'll go through that in detail. And then I'm really kind of interested. I've talked about this a couple of times before, but I want to present this to this CMS Philly audience in part 3. A very general thinking about, especially with this crisis right now, what is the future going to be in terms of a knowledge-based economy? And in particular, I think that most people, and this is what I've been working on with all of these knowledge systems I've been developing over the years, is to create an economy where people essentially get paid to do knowledge research on the web, and they provide knowledge services in a future economy. And part of this will involve something I call the Internet Homestead project. I'll tell you more about that in a minute, but that's where I'm going with this talk, to go cover those particular things. Okay, so I have a PhD in biophysics from UC Berkeley. I now am doing neurobiology and mathematical modeling of biological systems, and in particular, molecular genetic control of neural development. I was at Cornell Medical School for about 16 years. I'm still interested in mathematics, and in particular, I'm very interested in programming languages and using them for computational analysis of DNA transcription codes, which are the pattern language in the genome for programming cell-specific expression. Every cell in your body has the same set of genes, the same genome, but some cells turn on genes that make them become neurons. And how does that work? What is the molecular mechanism that controls that? That's what we're going to talk about first. So I'm a geneticist as well as a genomics researcher, and as such, I work on a model organism called fruit flies, and it's a very powerful model organism. I'll show you some things that are just amazing that you can do with fruit flies. So what I discovered, along with others, is that there's a set of what are called helix-to-helix transcription factors that control nervous system development, neural precursor cell determination in the embryonic peripheral nervous system, or PNS. So this first panel on the right says control. That's the wild type or normal animal. What you're looking at, these little dots are neuronal nuclei that have been labeled with a molecular marker, basically. And what you can see, there's a very precise pattern of neurons that are repeated segmentally. But in this mutation down below, a specific subset of those neurons, it's called the Ahq deletion, a specific subset. Actually, Chris, can they see my pointer? Does anybody?
Yes, yeah, you can see your cursor. There is a little,
okay.
There should be another tool if you want to use a highlighter or a pen to circle next to the.
No, that won't be necessary. I wasn't clear to me whether they could see what I'm pointing at, because it'd be very hard for them to follow.
Sorry, I couldn't mute myself fast enough.
No, that's okay. Thanks. Okay, so anyway, historically, there's been a number of mutations studied that control nervous system development, make abnormal nervous system. So here's one. It's called Ahq. It's called a deletion. Deletion of the 4 HLH genes removes a specific subset of the PNS. This is a gene that removes the entire PNS. So all of these cells up here, this blurry thing here is the CNS that's out of focus. It's down in the middle. This is right on the surface. All of those peripheral neurons are missing. And this is the gene that I cloned when I was a postdoc many years ago. And it turned out to be, remarkably, a previously identified sex determination gene called daughterless. And this was a huge surprise at the time, but and people didn't believe it, but it's now like textbook stuff. So daughterless is probably the best really well-characterized multifunctional gene that controls 2 apparently very different processes, sex determination and cell determination. And it turns out, you may know that humans only have about 23,000 genes. That also was a huge surprise. And it's because these genes get used over and over in different combinations in a combinatorial system. So you have sort of like a factorial computational functional ability from a small number of genes. And so you have genes controlling sex determination and cell determination doing the same thing. So the HLH domain that defines this helix-to-helix transcription factor family is a dimerization domain. And the daughterless protein that's expressed from the daughterless gene forms DNA binding heterodimers with each of the 4 Ahq proteins. And all of the information that I told you is conserved from Drosophila up to humans And so these are actually cancer-forming genes, so they get mutated in humans. But the fly has been an incredible model system for studying nervous system development, and it has a lot of implications for cancer as well. Okay, so one of the incredible value or potential of flies is there's been 100 years of genetics and many mutations that affect nervous system development. Now, the hairs that you see here are called chetae, and those are sensory structures that are innervated by neurons. So there's a neuron underneath each one of these that's sending a process into the CNS and sending sensory information. So all of these hairs are the progeny of a single neuronal precursor, the neuron inside and the hair outside. And when you see a missing hair, you're missing a neuron on the inside because that's part of the process. So here's a mutation called Ahq that removes, chetae is Greek for bristle. Ahq is missing some bristles. There's another mutation here called hairy. It's got a lot of extra bristles, hence the name hairy. In particular, there's a whole bunch of bristles on this wing, the second vein in this wing. And I'll show you more about that. But what was interesting is, and here's another mutation that has almost the exact same phenotype, but it's in a different gene, and it's called hairy wing. So these are actually mutations in HLH genes, all of them. Hairy is a repressor that genetically represses the Ahq positive HLH gene. And the question that I started working on many years ago when I started my own lab after cloning dollarless was, how does hairy work as a repressor? And it took us a while to figure out, but in fact, it turns out this is the Ahq gene. So every gene has a promoter. There's a cluster of transcription factor binding sites upstream, and Ahq has 4 class A activator binding sites and 1 class C repressor binding site. And if you mutate this repressor binding site, that's what hairy binds to. Okay, so here is a normal fly picture. This is a real photograph. Here's the hairy mutation that creates those extra hairs on that wing vein that I showed you in the drawing. And here's the mutation. If we just mutate this one DNA site in the hairy promoter, or sorry, the Ahq promoter, hairy can no longer bind here. And it's just like hairy was mutated. You get exactly the same phenotype. So this is the kind of amazing genetic analysis you can do with a model system like fruit flies. And in this case, you have these extra hairs because there's no hairy protein. In this case, hairy protein is there, but it can't bind to this site in the Ahq promoter. So one of the things we wanted to do when we discovered this was to ask the question, can we look for similar patterns of binding sites? So this cluster of 4 DNA binding sites, 1 repressor, 3 activator sites. Can we look in the whole genome? And that's what I call whole genome computational analysis. And interestingly enough, you can do this really easily with regular expressions, which I mentioned that in this talk because you're all a bunch of developers and you probably know about regular expressions. And I'm showing you how powerful they can be for this kind of genomic analysis. So here's a diagram showing that regular expressions are a pattern matching language capable of specifying virtually any desired combination of DNA sites, and also the architectures, not just what sites are there, but what their order is, the spacing, and all that kind of stuff. All of that can be written into a regular expression. And then you can, this is where all that information has been put together in one line. You basically, with a single line of code, can search for any desired pattern of DNA sites in the genome, across the whole genome. So that's what I've been doing since about 2001 when the fly genome sequence came out. So typically what you have when you do that whole genome analysis or any kind of really large scale genetic or genomic analysis, you get a lot more positive results than you know how to deal with. So when we do that, run that pattern across the genome, we find about 10,000 positives, okay? So the question is, how do you decide which of these thousands of computational results to test for function? This is the classic genomics problem, getting too many false positives. So the way we do it, there's different ways you can do it. And one of the ways we do it that is really powerful is we visualize which of those clusters are sitting in the promoter of target genes in the pathway. And we ignore all the rest of them. So here's this is what's called a Genome Browser. We're looking at a drawing that shows the DNA coordinates up here. And then there's various genes that have been discovered, and they're displayed in these different tracks. This is a data mashup from several different sources of information. And this is the cluster of actually similar to the Ahq promoter that's sitting in the promoter of a gene called NOT that's involved in neural differentiation. So this allows us to throw away about 99.9% of those clusters that we saw up here that are some of which are functional, some of which aren't. But this allows us to just focus on ones that are in target genes of interest, and that really helps us to be successful. So what we're doing here is visualizing the Ahq promoter. I showed you a different picture of it here, where this is a sort of classic drawing where the promoter, there's it's these repressor and 3 activator sites. Here, this same thing is shown using the G clusters computational analysis. See the same thing, Ahq promoter. So what I want to show you now is how you can do really remarkable things with a content management system like Drupal to turn it into a custom knowledge base for doing really, really huge amounts of data analysis very, very effectively. So the program I developed is called G clusters, stands for genes with clusters. I'm only interested in the clusters that are sitting in promoters of genes. And I just showed you an example of the Ahq gene and the promoter where you mutate it, you get a hairy repressor binding site phenotype. So all of the information and images are processed and stored in a local custom knowledge base that's Drupal. And also we have developed an Ajax interface for dynamic real-time interaction with this. So I'm going to switch now to actually show you what G clusters look like. So this is the real live program. It's still running on my server. Here is the result of one particular analysis looking for one particular regular expression pattern that turned up about 675 positive results. And you have to decide which one of those are you going to work on. You don't want to work on too many of them because it's a huge amount of work, and probably most of them don't mean anything. So you come up with various ways of prioritizing it. And basically what we've done is we've used genetic information, I'll show you in a second here, from a genomics online database called Flybase to prioritize which of these many, many results are likely to be in the pathway we're interested in. So what you see, interestingly, is the number 4 here in this result is Ahq. So that's basically positive control. We found the one that we should have found. And let me pull this up again. Okay. Okay, so now this is a G clusters display of Ahq with, again, those same 4 binding sites upstream of it. And what we want to do now is look at some of these other genes. So for example, the next one in the list of prioritized genes is called NUR, which is short for neuralize. It's known to be involved in nervous system development. And let me show you this. So the way this interface is designed, so this is Ajax. Again, I'm storing all of this information locally in the Drupal database. And that way, it takes about 3 minutes to create each one of these images like this. You don't want to be sitting there waiting for that to be created while you're trying to do the analysis. You want all that to be stored. We have an Ajax, very quick interaction user interface with this. So I can just go through large numbers of these genes, and the way it's designed is initially it pops up with the gene open. You can click on it once and it'll move out of the way and reveal this other information. It's called gene function information, and it says automatically mined systematically from Flybase. So Flybase is a huge, it's like, I don't even know what it is now. The last time I checked a few years ago was 50 gigabytes of data. And it has all of this genetic information from many years of genetic analysis. It tells you what these genes are doing. And in particular, it tells you that it's expressed in certain things, and also that it interacts with other certain genes. So Ahq is shown to interact genetically with Harry. I told you that before, and it does that through that binding site. Okay, so this online or this local custom knowledge base that we built out of a content management system, Drupal in this case, allows us to do really large scale analysis and just filter out everything using using information from, there's called a Go ontology, that is the kind of information that's being pulled out. These are all Go ontology terms here. Okay, so you've got a domain-specific language you're using to analyze your data. Okay, so what I want to do now is go back to the slides, and these are, so I made screenshots of these things of G clusters that are in the PDF that I'll give you. So actually, I think I'll leave this slide out. It turns out that gene neuralized that I told you about has these binding site clusters similar to the Ahq promoter that look like they might be targets for regulating neuralized. Neuralized is expressed specifically in neural precursors. That's what this slide actually shows here, that this is a neural precursor, neuralized is expressed there, and it's being regulated by the upstream genes, in this case, Ahq and some other genes in this pathway. So what we were able to do is take one of these clusters that's like a little promoter, and there's alternative promoters on this gene. These are called introns. They're spliced out. Here's promoter 1, here's promoter 2. We took this one cluster and put it in a transgenic fly. So we can make new flies that have, whatever we want in it. And we put the just as 4 clusters or 4 site cluster in from the neuralized promoter into a fruit fly. So this is a transgenic fly, and what you see is it's turning on expression. There's a marker reporter gene here that shows where it's expressed, and it's expressed specifically in neuronal nuclei. So this has really important implications. The reason we're doing all of this is to try to understand how we could express genes in humans, in particular cells. Say we want to, for some particular cancer, express cells and neurons. This is exactly the kind of thing that we would do, make use of that. So those implications here are really interesting and important for this transcription family, the helix-to-helix transcription factors of conservation in humans, and the fact that they could be used for gene targeting. So what I want to do now is switch to talking about the use of Drupal as a content management system, knowledge management system, in a crisis project that I started and had a lot of help from other Drupal developers in 2010 after the Haiti earthquake. And the problem was to try to get food to people who needed it. There was a lot of food sitting on boats in airport and stuff like that. But the people who needed the food were out in the environs. There was a aftershock after about a week after the first earthquake, and everybody moved out of town into camps outside the town. So there was all of this food and stuff in the town and at the airport and so forth, but the people were all out in the surroundings. And so what we had to do was to find a way to get the food that people needed and other medicine and so forth to them. And what ended up turning out to be extremely powerful, this was right at the time that hashtags, Twitter was starting to develop as a hashtag system. And one of the people on the project, Greg Harvey, saw that Twitter hashtags were being used to label tweets from people who were, in need of being rescued or food and so forth. There were 15 million SMS or tweets sent out of Haiti in the first 2 weeks after the earthquake. And by using this hashtag system that was just coming into use in general, we were able to create this panels-based mashup where I'm not sure if you can read, this says pound Haiti, pound need, pound Haiti, pound have, and we'd have this panel here with people who need things, and here's with people who have things. So this is also another case where we took all this information, stored it locally in a Drupal database. We can now search all of these messages and prioritize them as to the size, the number of people who need help and where they're located and all that sort of stuff. And we were able to help the World Food Organization get food to people who needed it. So what was interesting to me was that it turned out, as I was looking back on this, that the same kinds of processes and technologies that were very useful for genomics were really useful for this crisis project. And I just want to mention that in addition to this really important work that Greg Harvey did, a Drupal developer named Patrick Teglia created this logo called the Haiti earthquake information, which is a really nice logo you can see there. So there's all sorts of design opportunities for crisis projects and coding opportunities and so forth. And I want to invite all of you to think about how you can now contribute to this COVID-19 crisis project. It's built on Panopoly. If you're not familiar with Panopoly, it was created about 10 years ago as the next generation for views and panels for D7. And Earl Miles, who was the originator of views and panels, worked on that specifically with Pantheon and some other people to really develop a powerful system for analyzing or creating Drupal sites. And because of this, as soon as I found out that it's the next generation of views and panels, that was pretty much all I needed to know. And I started using it. I've been very happy with it ever since. To my knowledge, Panopoly is still the most widely used base distro for other more complicated distros like OpenAtrium and so forth. And I would really recommend it to folks for use. So what I want to do now is talk about this COVID project. Genomics Knowledge Systems is the name of my consultancy for creating knowledge tools, and this is actually running on my own research site right now. But it's a Drupal 7 Panopoly based knowledge system, and what I want to do now is to show you a little bit about Panopoly and Drupal for this purpose. Okay, so here we have the real site, and I loaded into this intro page the same description for the session. What's the motivation for this project? We're trying to essentially store locally in a custom knowledge base the most important information that we can find about how to address this incredibly urgent crisis that's taking over the United States and is almost taking over New York City. I live in New York City, so this is of deep interest to me. The person who lives across the street from me for the last 20 years died on April 17th from COVID. So this is real stuff. Okay, so What I want to do is to invite other Drupal developers, and you don't have to be a Drupal developer if you're a design person, you want to design a logo for this, or help with sort of information architecture and that sort of stuff. There's a specific need for sort of a home page where I'll show that at the end based on the part of Drupal that's called the demo page. Has a certain kind of layout that I would like to incorporate into this, but I don't really have time to do that myself. So, and I'm probably not very good at that kind of thing. So there are collaboration possibilities for software developers. There's also a project, you may have heard that contact tracing is a major new next step for this antivirus crisis response, and there's a Node.js based contact tracing system that I'd like to integrate that's based on Node.js. I've done Node.js development myself for several years in addition to Drupal. So also I want to have frontline medical workers, doctors, nurses, and so forth involved in this as well. And also, so I'm a geneticist. There's other virologists, epidemiologists, and so forth I'd like to get involved. I've got 2 other geneticists collaborating with me now. So, and then also journalists, to have news journalists be participating. So this collaboration I see as something, as a academic scientist, what I want to, I'm not getting paid out of this. I've been working on full-time for free for 2 months, and I will work on it full-time for free for the indefinite future. But I do want to have some publications come out of this. So I want to have co-authors, and the website itself is a publication. Now, some of you may know that there's a module in Drupal called Book, that you can actually create a book out of a website. So, and there was I think I mentioned it down here. The book module was used to create a book called The Definitive Guide to Drupal 7. And we can do a similar thing here. So one reason I like Panopoly, I have found Panopoly to be a really useful sort of intuitive go-to responsive design web-based content management system. And let me show you why. So you have a bunch of tabs up here. There's a site outline at the right-hand side here that is essentially like table of contents for a book, okay? And if you collapse, the reason I put the site outline on the right is if you collapse this down, you have these menu tabs collapse nicely, and you have the body of the page, but down below it is the site outline. So you can navigate to any site in the database just from this very I think elegant and sort of universal and real easy to sort of wrap your head around. People, it's kind of intuitively obvious how it works for most people. So that's one of the main reasons I want to use Panopoly and why I've been using it, as well as some other things. So again, if we start looking now at, I'm going to go back to my slides now. So this project has gone through, in the last 2 months, several phases, about 3 phases, in terms of who it's trying to focus on most to get help. And in this 3rd phase, since about March 17, it's been focused on the hospital ER workers. These are the emergency room workers. And by ER worker, I mean all of the people associated with that hospital. It might be the intensive care unit upstairs, people in the ER on the ground floor, the ambulance people who are bringing in new cases, and so forth. But the design strategy between this whole project from the very beginning, and it's turned out to work really well through these 3 phases so far, is it was clear right from the go that there's a very limited amount of testing kits available. And that has kind of defined how this project has evolved. And also, if you've been paying attention to this crisis at all, it's still like the major problem that there's not enough testing. And for example, you can't do contact tracing without testing. So what we're trying to do right now is focus on this really, really urgent need of the hospital workers to be tested. Most of these hospital workers have not been tested yet, even though, and they're dying, okay, because they don't have enough also personal protective equipment, PPE it's called. So here is, let's just go through a couple sites here. Okay, so this is a page called Why Microtesting and Microtracing, i.e. microcontainment of the New York City hospital ERs is critically important at this time. So there's an image here. This is what motivated all of this about, you know, mid-March, after 2 other phases of development. These stories started coming out of worker dies from coronavirus at New York hospital, where nurses wear trash bags as protectors. That's the title of this news article, okay? So this is an active link to that article, and you see the guy who, there's a guy who died, and the people in this article are saying that they're blaming the hospital for not providing this guy with enough protective equipment. So the lack of personal protective equipment, PPE, is just a huge problem in this whole thing, especially in New York City. So what I'm doing in this site is trying to put together these really critical bits of news information, you know, using the news and other sources of information. I also read the literature about virology, you know, how this coronaviruses works and that sort of stuff. But a lot of it has to do with just following the news very closely every day, and it's changing. Every crisis project is changing so rapidly that it's almost impossible to follow. And that's one of these key ideas of this content management knowledge system of locking this information. You know, you stop this river of news and it's just flowing by constantly. You figure out what are, you figure out what are absolutely the most important things.
Michael, just to interrupt first.
Yeah.
We've got about 3 to 5 minutes left.
Well, okay, we started about 15 minutes late.
Right, I know. I waited to make sure you had enough time. So let's say it's a 5-minute warning.
Well, okay. I'm just saying again, we started 15 minutes late because I mean I was there. At any rate, I will try to finish this up. Okay?
Yeah, we have till the top of the hour. I just want to make sure that we're done by 4. That's all. Thanks. Okay.
Well, I'll just say one more time. We started 15 minutes late, but at any rate, I will be done in 10 minutes. Okay. So all of the slides that I've shown you are in a PDF, and there's actually, if you follow these links, if you go to the website and you follow these links, you can actually find all of the genomic stuff. It's under research down here at the bottom. Even including G clusters is running on this website. So at any rate, let's go back to this. So basically, I'm back on the website now. This is a page that we're talking about. This worker dies from coronavirus at this hospital. Another very important bit of news around this time was the fact that these deaths were starting to really add up there was a news article called 13 deaths in one day at the Elmhurst Hospital in Queens, okay? And that really caught my attention as well. It turns out that on April 6th, the same hospital, there were 20 deaths that day. So, you know, this is these hospital and the hospital workers themselves are starting to die. Okay, so the let me just tell you here. So the focus of this is, again, on these ER workers, the doctors, nurses, EMT, police officers, and so forth, all these people who are working. They should be prioritized for being tested, and they're not. There was a news report on April 3rd that 25% of the EMTs called in sick, 25% of fire department called in sick. The next day, 20% of the New York Police Department had called in sick. In April 10th, there was an interview with an ER doctor in a hospital in the Bronx named Ernest Patty, who had mentioned that a friend of his, a colleague, a doctor, a trauma surgeon named Ronald Verrier had died himself from the virus. And in a follow-up article or interview with the same doctor from the same hospital just a week later, April 17th, he mentioned that he had lost 4 additional hospital colleagues. Okay, so this crisis is just taking over the hospitals in New York City, and if we don't get this critically needed protection and other and testing in particular to these medical workers, they're going to get infected and they're going to die, and that virus can take over. Basically, I mean, it's widely acknowledged that if the virus wins, it's going to win first by destroying the New York City hospitals, then New York City and the economy, New York State, and potentially spread throughout the U.S. Now, if you're following this at all, you know that there's many, many outbreaks throughout the United States now because of this very, very contagious spread of this virus, which is also addressed in some of the pages on this website. So again, the design strategy was to try to make absolutely the most efficient use of the testing kits. Right now they're doing like drive-in testing of random people on the highway who, they call and they say I've got symptoms, but they're not doing it in any sort of organized way. They should be using those tests to test all of these ER workers, and they say that they say they're supposed to do that, but it hasn't been happening. It still isn't happening. Okay, so I told you about that. Here's the guy who died. So there's one other sort of story I want to tell you about. So there was a news article about this 13 deaths in a day. I mentioned that. I said also that there's 20 deaths in that same hospital. This woman, Dr. Colleen Smith, gave a video that is this link right here on this article, where she took you through the hospital and she showed us a lot of stuff. And what she said, this is from a transcript, she said that 10 residents and also many of our nurses and attending physicians are sick. I want people to know, this was on about March 25th. I want people to know that, we're getting exposed over and over. We can't get testing even if we have symptoms. And that's still the case, that even though we're now like 3 weeks after that, it's still the case that the doctors themselves cannot get tested, and they don't have these protective masks. They're called N95 masks. She said that she puts one on. They're supposed to, every time they see another patient, they're supposed to throw out their previous N95 masks, but they have to use them. There was an article or a report of a nurse in California who was told to wear her N95 for a month. And I've seen many, many reports where people have been told to use them for a week. Okay, so there's this really urgent need for personal protective equipment and for testing of the doctors. So I started this additional project on the site. It's called Emergency Protocols for PPE Decontamination. As a molecular geneticist, I have the kind of background where I could come up with this thing. So you could read that if you want. Some good news is that I have been able to contact the director of ER at Elmhurst Hospital, where Colleen Smith worked. And his name is Stuart Kessler. We had some discussions about them using these emergency protocols, and he's forwarded the information to other people on his staff, including to Dr. Smith. And the plan is for, now that I've made that, established that content, is for Elmhurst Hospital Group to become a C group for a New York City-wide hospital ER worker group collaboration, using both the knowledge and tools provided on this crisis policy knowledge base, integrated with the digital contact tracing tools provided by Demagi. Demagi also has a lot of very powerful tools for supply supply chain ordering and so forth. So I just want to show something here, and I'll mention it sort of in passing. There was one family in North Jersey, the Fusco family, where they've already had, this was like a couple of weeks ago, 4 dead and 3 hospitalized because people didn't know how to take care of these people. One of them went to the hospital. When she was sent home, she infected her whole family. And, you know, half the people you see in this picture are already dead. Okay, so this is an important story and lesson here that needs to be told. It's the fact that we're not doing testing and we're sending people out of hospitals who are contagious that has resulted in a huge amount of death in New York City area and North Jersey. So the last thing I want to talk about in terms of Panopoly is there is what's called a demo page that lets you create various sort of layouts of news and other things. They have like featured links and so forth. This is something I haven't spent any time on. I haven't actually seen any documentation of it. There probably is some, but I actually would really like to get some help with this. If someone can, you know, you don't have to be a Drupal developer, but just, you know, come up with what seems to be the best kind of design layout to tell these stories. Okay, there's a lot of really good information on the site, but it's spread around in different places and it's hard to see the big picture. So finally, this is just the same slide I started with. We're now on the last part. I kind of thought about this talk for CMS Philly in terms of all of you are CMS developers, or you wouldn't be attending this, okay? And I've tried to show you how you can take a CMS. I happen to use Drupal, but there's many others. And you can build a custom knowledge-based system. And I've been thinking a lot about how do we move toward a knowledge-based economy. And it's even more important now that we have this lockdown where people are just going to have to come up with different ways of making money. And one of the ways that I think is going to be necessary is for people to become knowledge experts in some knowledge domain. Everybody should not just be a developer, but should have some particular knowledge expertise. And one project that I thought about is something that I call the Internet Homestead Project, where you're probably familiar with the Homestead Act that President Lincoln started in the Civil War, where basically anybody after the war could go out west and homestead, you know, 5 acres of land or something like that. And if you improved it, you could keep it. So what I'm proposing is to give everybody a copy of this Genomics knowledge system where they can create their own, you know, custom knowledge base or whatever they're interested in. People who are, you know, collaborating on the project, there will the plan is to have a book come out of this, and maybe some research articles too, like on archive.org. But for sure, a book about this particular project, but also a more general knowledge system based on well, Genomics, but based on Panopoly and some other things like Node.js. So that pretty much, excuse me, wraps it up. These are my colleagues that I've been collaborating with over the years, and I'll take questions now.
All right, I don't think we have any questions from anybody. Michael,
Okay.
Thank you so much.